Hi, this is Mrs. Stram here. I am here again to tell you guys a little bit more about the Amgen Biotech experience. I'm going to be going over Lab 2 so that you guys know what Lab 2 is about. So, in Lab 2, we're going to be working with two plasmids. Just a reminder, plasmids are circular pieces of DNA. And circular pieces of DNA, like this plasmid, can be used to transform bacteria cells. In other words, introduce a new gene or new genetic material into a bacteria cell. So we're going to be working with this plasmid right here called PCAN-R. I'll tell you a little bit about that. The P in both of these represents the word plasmid, talking about the ring of DNA. The CAN part of this represents the fact that this plasmid contains kanamycin resistance. Kanamycin is an antibiotic, so if you give a bacteria cell this piece of DNA, it can survive in the presence of the antibiotic kanamycin. The R here stands for recombinant because this is a recombinant plasmid. This recombinant plasmid contains new genetic material, which in this case is this portion. And specifically, this RFP gene stands for red fluorescent protein, which was found and isolated from a sea anemone from Dr. Shen in the San Diego labs, who won the Nobel Prize for his work with fluorescent proteins in 2008. This plasmid of DNA is known as a cloning vector. Okay? And a cloning vector simply means that this plasmid is able to copy the DNA through DNA replication and make hundreds and hundreds and hundreds and hundreds and hundreds of plasmids if it is put into an E. coli cell. The E. coli's DNA polymerase will make lots of copies of it so that you get millions and millions and millions of these plasmids. That can be useful if you want to make a lot of copies of this gene right here. Once you have a lot of copies of this, because you've used the cloning vector, you can use this plasmid. This plasmid, the P-ERA plasmid, is called an expression vector. Okay? And it's called an expression vector. Expression because that's referring to the fact that the protein is expressed if it is put in this complex. Notice over here, instead of having a kanamycin resistance gene like PCAN, this one has an ampicillin resistance gene. So once we put this plasmid into a bacteria cell and try and grow it on a plate or a petri dish, it will survive even in the presence of the antibiotic ampicillin. This structure over here is basically a molecular on and off switch that controls gene expression. So I'm going to be explaining right now then why we call this an expression vector. If we take the gene that we've cloned, we've made lots and lots of copies of this, so we have a whole bunch of RFP, the gene, we can then use molecular scissors to cut this gene out, and we can glue it over here in this section so that we create a plasmid that has a gene we want made, as well as an on and off switch for DNA, not DNA, but RNA transcription, okay? So P-ERA, the ERA is simply standing for this ARAC gene, which codes for the ARAC protein, which we'll talk about later, and it controls whether or not the gene is turned on or off. So let's clear some of this stuff up so that we can see what's going to happen. Lab 2 then, I've introduced these plasmids to you now, so you know this one over here is a cloning vector, makes lots of copies of DNA. This one over here is an expression vector, is used to actually do transcription and translation so that you make a protein structure. Lab 2 is a process by which you do a restriction digest, which means you add restriction enzymes. In this case, we're going to be adding BAMH1 and HindE3, and we're going to use those restriction enzymes to cut DNA. So we're going to cut these two pieces apart on each plasmid, just like this. That purple represents BAMH1, the orange represents HindE3. It cuts. Same thing over on the P-ERA plasmid, HindE3 and BAMH1 cut. And later, we're going to be combining this fragment with this fragment. Okay? If I go to the next slide to show you guys, on this slide what you can see is the 4 linear pieces of DNA. I'm calling them linear because they're now basically just a strip of DNA that is not connected to itself, so it's no longer a circle. And we're going to want to combine this piece of linear DNA with this piece of linear DNA so that we have a gene expression on-off switch accompanied with a protein